Okay. Well, it's wonderful to be here, and as you'll see, today's lecture will be somewhat a change of pace from the classical statistics I was talking about last week. So, one of the topics of the school is Evo-Devo. Okay, that means evolutionary biology, the evolution of developmental biology and developmental systems. And I'm really a developmental biologist by training. I mean, and also a theoretician. I sometimes say I'm a molecular biologist who isn't afraid of mathematics. And so what I want to do is introduce you to some big questions, number one, and I know that a lot of people here are coming from a theoretical or an informatic background. So I'd also like to give you an introduction to the messiness of biological reality, because if you're making theories about the natural world, you have to know what's out there in the natural world. And one of the things that unites a lot of what I'm going to talk about in the three lectures is an idea that was proposed a long time ago by a British biologist named Conrad Waddington. And what Waddington was interested in was a very fundamental problem, which is, as you look over the record of evolution, okay, what's happened, it's clear that organisms adapt to their environment. The ancestors of whales had legs, but when they started living in the ocean, they lost their legs. They adapted. They adapted to the aquatic environment. And this is sort of a commonplace to anyone who goes to natural history museums, just if you know about evolution at a popular level. But there's a big problem that comes up, which is if you think about adaptation over evolutionary history, it means that organisms and their genes have developed new functions to do new things to be adapted better. And you can see this in popular culture, for example, in the movie Godzilla, okay, where the radiation causes mutations and somehow you get this gigantic fire-breathing monster. Except if you're working in the lab and you make mutations, you don't get fire-breathing flies, you get sick flies. And in general, most of the mutations that you see are damaging. And so this is a really fundamental problem. And if you talk to critics of evolution, for example, there exist some of them in the United States, I don't know about India, this is a point that people bring up a lot, which is that, you know, how can you talk about adaptation and evolution when all the mutants you make in a lab are sick? Okay, so this is the problem that was taken up by Conrad Waddington, because if you have acquired, the old idea from Lamarck was that acquired characteristics could be inherited. So the obvious example is the giraffes, they're reaching up to eat leaves on the trees, and the giraffes that have the longest necks do the best. And so they have more children and they also have longer necks. Okay, genetics showed that that was wrong. And so, So, you know, I've given out some reading material, and if you don't read any of the reading material, take a look at this paper by Waddington. It is amazingly short. It is really a natural history paper. But in the raw material of evolutionary theory is the writings of naturalists, because that's the data, what happens out there in the natural world. Now, in some sense, the problem posed by Waddington is still unsolved. And so there's also a sense in which I'm going to be sort of pecking around the edges. I'm going to give you a couple of results that touch on this problem, but I think the core problem is still unsolved, and I want to bring it to your attention because maybe some of you will go out and solve it. It's also a problem that has to do with development, because although a lot of the, it's easy to invest, it's easier, not easy, it's easier to investigate evolution in one-celled organisms such as yeast or bacteria, the fact of the matter is the reason we're interested in evolution in the first place is that's how the whole set of life on Earth evolved, okay? And that includes large animals and plants. They all develop. They're multicellular. So ultimately, that's a problem you have to come to grips with. And in order to do that, you need to know some of the details of the process. So I'll be talking a little bit about development this afternoon. Development is controlled by genes, and so the first question you start to ask is, how do the genes get turned on and off? And by the way, just like last week, okay, this is not a seminar designed to snow people. This is supposed to be a school. So I'd like to encourage all of you, since you come from very diverse backgrounds, to stop me with questions. And there's some plasticity in the talks. I get to speak 3 times, so if things spill over to the next one, you know, so it goes. So I want to start today by talking about one single paragraph in Waddington's paper that has been tremendously intellectually fruitful over the last 6 decades, and not just in biology. So from a particular observation, okay, that I'll talk about this afternoon, and I'll come back to this quote, Waddington said, developmental reactions, as they occur in organisms submitted to natural selection, are in general canalized. That is to say, they are adjusted so as to bring about one definite end result, regardless of minor variations during the course of the reaction. Now, this is a particularly remarkable statement because at the time it was made, no one had any clue what these developmental reactions might be. And furthermore, the role of DNA as the genetic material was as yet uncharacterized. And also in the afternoon, I'll get back to some of these issues about Waddington liked to make drawings. And so, for example, he illustrated canalization by this idea of a river running through a landscape. So, for example, in one of my discussions at dinner, you know, I mentioned Waddington, and one of the students says, oh, yeah, isn't he the landscape guy? Well, sort of, okay? We'll come back to that in the afternoon. Landscapes are potentials for you theoreticians, and there's a lot that you can't do with potentials. So what I want to talk about today, okay, and another word for canalization, okay, I'll use the board as needed, is that paragraph by Waddington about things being directed towards one end result has another aspect, which is buffering of phenotype from genotype. Okay, and that's something that's based on something that you see all over biology, where you can have large variations in genotype, relatively large, and you see the phenotype stays relatively constant. Okay, now actually let me, I'm just going to go make a couple of comments, and then I'll go on to that next slide. So in general, if you look out on people who work on canalization, they usually think of this in terms of canalization in terms of connections between different genes. That's what I'm going to talk about in the afternoon. Okay? But I called this talk Canalization in Cis. Okay? And for those of you who are not geneticists, I just want to give you a little bit of vocabulary. If you're talking about genetic actions that happen within one gene, okay, that's said to be cis. If they involve different genes, that's said to be trans. And so the general phenomenon I'm going to talk about today is that when you look at the control of genes, in multicellular organisms, they have very, very large control regions. And in fact, changes in this control region are extremely strongly buffered. And so my goal today is to give you one example of such a buffering. Now, in one sense, this is just the most elementary sort of theoretical concept. It fits into stuff we talked about last week. It's just an application of the law of large numbers, okay? But you can't look at this problem in terms of just sort of plain classical statistics. You have to know a lot of the features of the system in order to do that. And furthermore, in order to do it, it touches, so a lot of fundamental problems are wrapped up in this canalization idea. Okay, so here's a fundamental problem. You're all aware that the genetic code was solved a long time ago. I mean, it's almost 70 years. Okay, but what is the genetic code? The genetic code is the triplet code for proteins, and it's simply a set of relationships between sets of 3 bases and amino acids and the canonical 20 amino acids. It's a completely one-to-one mapping. It's sort of like the ASCII code for Western languages. This set of bases spells out histidine alanine. This ASCII, grouped in sets of 7, says hi. And in some sense, this is very straightforward as a code. Now, that's only half of what you have to know to understand how DNA codes for the structure of an organism. Because in the course of development of an organism, you have lots of different cells forming. The cells are different. Skin is not the same as brain or liver. And what's different about them is they express different genes. And that's a consequence of the fact that the genes are turning each other on and off, but there's a copy of the same genetic material in every cell. Okay, great. In order to construct an organism, you also have to know how the genes are turned on and off. Okay, this is a very peculiar problem. It is qualitatively different than the code for protein structure, because what you have is a case where various sets of bases, okay, and typically they tend to be binding sites for proteins that stick to the DNA. Okay, there's other things going on that this binding triggers, including spatial rearrangements. But in general, proteins bind to the DNA. This is a binding site for a fruit fly, a Drosophilid melanogaster gene called hunchback. But what it's doing is very subtle, because it's saying the gene must be on in the correct cell type and off in the correct cell type. So here, instead of a one-to-one relationship, this series of bases specifies many combinations of transcription factors, not a single amino acid. Okay, so there's already a very, very tricky aspect in this. I must point out that the cis-regulatory code in bacteria, okay, and Mukund Patai alluded to that in his lecture last year, is actually much, much, much simpler. Okay, it was worked out in about 5 years by 2 brilliant Frenchmen, Jacob and Monod. But bacteria are very complex. They don't differentiate, and they don't have this problem of forming cell types. Now, I have to also tell you about a little bit about the experimental organism that we study this problem in. Okay, so I work on fruit flies, Drosophila melanogaster, and now other species. So I will eventually be talking about multiple species of flies. And this is a great system for working in because the early embryo is exceptionally simple. Okay, now I don't want to leave you dazed with too much biology, but what you should keep in mind is that during the very early phase of the development of fruit flies, the embryo looks like a hollow American football. Okay, so this is approximately an ellipsoid, and inside this football, there is a shell of nuclei. Okay, and flies are very specialized for rapid development So there's some other special things about them, okay? I won't say all of them right now, but one of them is that the nuclei are not separated by membranes. You can essentially treat these embryos like microarrays, that instead of having to pay some company for it, mother fly provides for you as long as you provide her with a little bit of cornmeal and molasses and yeast. Okay, now I work on a particular system of genes that are responsible for making the segmented pattern of the embryo. And I'll return to that issue of the developmental role of those genes in the afternoon. But for the morning, I just want to use them as a test system for understanding the cis-regulatory code, how the genes get turned on and off. Now, what you need to know for today is these genes fall in 4 classes, okay? The segment polarity genes are the final output of the segmentation system. I'll say a little bit more about them in the afternoon. There are initial conditions provided by the mother, okay? And when I say provided by the mother, I mean these eggs are not simple things, okay? They have all kinds of internal asymmetries when the egg is laid. Okay, for the purpose of making the fly segments, there's 3 genes that have message put into the embryo by the mother, bicoid, caudal, and hunchback. This shows bicoid. Okay, there's several cell divisions that happen during this period. This is after the 11th division. And you can see each one of these dots is a nucleus. And these embryos have been stained with antibodies to these particular proteins by a process I won't get into here. It's essentially some very effective biological black magic. And you can arrange for these nuclei to fluoresce in a manner that is proportional to the concentration of a particular protein in the nucleus. And when I got into this, you could make these measurements in a clean way, essentially only in flies. Okay, this technology works in all kinds of tissues and organisms now. Now, the zygote. Early in the blastoderm period, the zygotic genes turn on, and there are 2 classes of genes that act in the blastoderm, the gap genes and the pair role genes. So I'm not going to talk about mutant phenotypes very much. Suffice it to say that these names, like the names of all genes in flies, come from the mutant phenotype. So for example, a gap gene, if I was a mutant for a gap gene, I would be missing a part of my body about this big. Needless to say, those mutant mutations are fatal. And that's another difference, by the way, between the point of view of developmental geneticists and population geneticists, okay? So we had some excellent lectures last week from Kavita, and she was talking about situations where the composition of the alleles in a population change under selection using approximations such as additivity, okay? And developmental genetics is sort of the opposite of this. Because when you look at these genes, they are in a supremely non-additive situation, okay? In the sense that if you deactivate one of them, it's lethal. And if you're lucky, it's lethal in a way that is scientifically informative, okay? And so one of the things I'm interested in is bringing these 2 perspectives together. But the tension between these perspectives is something that you have to keep in mind when you look at developmental or evolutionary problems. Yes?
Multiple cells, is this a single cell with many nuclei, or are there multiple cells here?
Well, it's called syncytial, so that technically means a single cell with multiple nuclei, but that's wrong, okay? Because essentially it's a situation where there are multiple cells that are not surrounded by membranes because there's this cytoskeletal apparatus around each nucleus And it's, in that sense, very different from a classic syncytium like a muscle fiber, which is basically a big homogeneous tube with multiple nuclei. This is not homogeneous. One of the great simplifications that won't affect this morning, but will be very important this afternoon, is it means that you don't have to worry about signaling between cells. You can just allow for these transcription factors, which is what all of these proteins are, to diffuse from nucleus to nucleus.
So what the first picture is showing is that BCD has a high concentration on the left and is a declining concentration towards the right.
Yes, and you know, I forgot to say, yes, that's absolutely correct. And I should add something else that I forgot. All of these embryos are shown in an orientation with anterior to the left and dorsal up. So bicoid is high in the anterior and it falls off to the posterior.
So the nucleus itself is the place where it's most expressed, right, this protein? What about the region surrounding the nucleus?
Well, of course, it's translated in the cytoplasm surrounding each nucleus, because that's where the ribosomes are. In fact, it's a DNA-binding protein. So it then goes back into the nucleus. And all of the proteins that I'll be talking about are DNA-binding proteins. They are so-called transcription factors, which means their binding to the DNA affects the transcriptional rate. So these are exactly the kinds of questions that we need. So, you know, that was asked by a professor, but you students shouldn't be shy. Yes.
Of the nucleus receding, so the dot on top seems to be much larger than the size subsequently.
That's essentially an optical illusion. The volume of the nuclei does increase a little bit during the S phase, but what you're actually seeing, oops, pardon me, what you're actually seeing here is that during this earlier period between there's 13 nuclear divisions during this stage. Earlier, the nuclei are kind of squished, and later they get more columnar. So the key point for what I'm going to talk about this morning is the segment pattern of the animal. Okay, insects, you might have noticed, are made of repeating units called segments. I'll show you a picture of a grown-up animal this afternoon. But the first manifestation of the repeating pattern of segments is the expression pattern of these so-called pair rule genes. So I was telling you about gap genes. They're zygotically expressed in large bands, about 10 or 15 nuclei wide. Here's a list of them. And the pair rule genes, which initially are not expressed in a striped, are initially not expressed in a striped pattern, are the first manifestation of the periodic pattern of segments. Okay, and in particular, even skipped, one of my favorite genes, is particularly important in that regard because it is the only pair rule gene that receives input only from gap genes. Okay, the other pair rule genes to various extents also take both gap and pair rule inputs. Okay, a lot of times when people talk about these, they show some diagram with boxes and arrows. And I'm not showing you that because I think these diagrams are less than helpful. Because as soon as you try to make them accurate, they start to get this hairball appearance. But what you should keep in mind is periodic pattern of even skip made from non-periodic pattern of gap genes. Now, when you ask how that happens, it turns out that it is an amazingly complicated problem. Okay, because the actual message from the even skip gene is only 1.5 kilobases. But the whole gene is 16 kilobases. And the part of the gene that is essential for making the 7 stripe pattern is still about 14 kilobases. All of this is biologically essential DNA. And so, in fact, the coding region of the gene is very small compared to the control region. And for those of you who are interested in things like statistical genetics and polymorphisms in human populations, most of the medically important SNPs, single nucleotide polymorphisms, are not in coding regions of genes. Okay, so if you do something like GWAS, okay, I'm not a statistician, even though I talked about statistics last week. One of the frustrating and unsatisfactory things about it is you have to use general purpose statistics because you have no clue how these bases actually affect the functioning of the gene. Yes?
In the last slide, is those photo related to genotype aspects or phenotype aspects?
Well, this is essentially in the middle of executing the genotype-phenotype map. Okay, so ultimately, I mean, you could call this a molecular phenotype, but ultimately the genes do things, and eventually you get a living, crawling fly. Yes, this is part of the phenotype, okay, because the phenotype is a 4-dimensional object in a developing organism, because it's not just the animal you see at one moment, okay, it is the whole process in time by which the animal develops. Okay, so getting back to this really complicated gene, you'll notice this yellow here, okay, this is the actual transcript. It has one intron that's not shown, okay, just like Mukund said the other day, tend to illustrate that with this little arrow. And then you'll notice these little red bands, MSE3, MSE2, FUTS-like, stripe 4 and 6, stripe 1 and stripe 5. What do those mean? Okay, those are things called enhancers. And so when people realized these genes were so large, okay, and by the way, you know that it's 16 kilobases because that's how much DNA you need to rescue a lethal mutation so that they live. Okay, so when people started to analyze these large genes, what they did was they took fragments of DNA and they linked them up to a completely inert physiological. Gotta watch these things. That's why I. Sorry, VJ. That's okay. I'm not going to worry about the screen. You take a fragment of DNA. And link it up with a so-called reporter. And the idea is that this construct is like a pH meter, because you could monitor what transcription is being driven by the LAC-Z gene, by the GFP, but that gene is physiologically inert. And that's obviously a very important thing if you're doing development, because since these proteins control how the animal develops, if you were to express some protein in an abnormal pattern, it would be apt to be fatal. And so let me come back to this big diagram. Okay, I'm actually moving a little bit slow. I hope it's not too slow, but I'm judging from the question. Yeah, you know, I can adjust the rest of the talk, but I think you should be oriented to sort of the actual things you contend with experimental data. Okay, so what these red stripes are little enhancers, okay? And what an enhancer means is if you take this fragment alone, okay, MSE3, it drives stripe 3. And actually, MSE3 also drives stripe 7. Okay, MSE means minimal stripe element. MSE2, which is the smallest piece of DNA that will drive stripe 2, is one of the best studied enhancers, and I'll have a lot to say about it. Okay, so the point is the action of the whole gene, in some sense, is the consequence of the action of these different enhancers. Okay, unfortunately, and I may or may not have time for this, but a paper talking about it is on the reading list, they're not really additive. They have some interactions. Okay, so this is the kind of thing that we need to work with to understand the cis-regulatory code. And this is the kind of data we have. Now, this is also another reason I work with flies, the fly embryo. They're so beautiful. And this shows giant, one of the gap genes in blue, purple in green, and Eve in red. Okay? And this is just to make the point that if you want to study this problem, you absolutely have to have data from individual cells at cellular resolution. Okay, there's a lot of beautiful work in functional genomics involving really elegant techniques, but the techniques involve grinding up cells. Okay, single cell stuff is coming along very nicely, but you would not want to work with this in terms of single cells, because if you ground up one of these embryos, you'd lose all the important information. Okay, you'd conclude that giant and Eve are expressed at about the same level. And of course, you're losing a lot of critical aspects of the system because they're not necessarily expressed in the same cells. Okay, the cis-regulatory genetic code is executed in a cell-by-cell basis, and you have to look at it that way. Okay, and the other thing that's really important is you need to have both inputs and outputs. Okay, and if you look around the experimental biology literature, that's beginning to happen in more and more cases. You can have with transcriptomes and proteomes, but classically it's often the case people doing genetics, they manipulate the genes, but they don't know what's going on with the regulators of the genes. So for example, the history of the protein structural code is one of the places where, theory was excellently deployed in biology by people like Francis Crick, but the final deciphering of the genetic code for structural proteins required some brute force, okay, where Marshall Nirenberg, by brute force, synthesized all the possible codons, did the reaction in vitro, and looked at what came out. Okay, so as yet, you cannot study transcriptional regulation in vitro, okay? The reason for that, by the way, is you may have the impression that chemistry is well understood. That's not quite right. Chemistry is well understood in well-mixed solutions, okay? And well-mixed solutions provide a good approximation for bacterial transcription, but in the case of multicellular organisms, the chemical reactions that control transcriptions are vectoral. They involve the spatial arrangement of proteins and chromosomes, and you cannot as yet reconstitute them from pure substances in a test tube. So you have to look in vivo, okay? So having inputs and outputs is now something you can get in tissue culture systems. Some of the techniques I'm going to talk about have now been applied to mammalian systems from tissue culture data, but we started out in embryos. I gotta watch these buttons. This one will turn it on. Yes, I see, I press it twice. My apologies for difficulties here. And you need a way to compare inputs to outputs. So because you're getting this data out of an in vivo system, you need to have, you need a model to compare, okay? Because this is obviously very complicated. one of the results, one of the reasons that this school is happening, and one of the results of the great explosion of technology in biology is that, you know, the experimental biologists can't always understand the results of their experiments, okay? They're probably, if any, are, you know, sometimes people bristle at this, but it happens. So you need to be able to calculate. And in order to construct a model, you need three things, okay? Two of them are very straightforward. You need to know the DNA sequence of the regulatory region, and you need to know something that we'll hear about later in the week, which is you need things called positional weight matrices. Okay, so I notice the way this course is arranged, you're getting hit with a lot of stuff kind of in parallel. Okay, simply take it from me that positional weight matrices are a way to predict how tightly a protein will bind to the DNA from the DNA sequence. Okay, they're not perfect in doing that, but the fundamental principles of how to get them are reasonably well understood. And a lot of the time, if you're not happy with your PWMs, the way to improve them is to get better data. And the really important thing, which is not science so much as an art, is C, informative experimental problems, okay? That's something that you have to learn to find yourself. Okay, now, you need quantitative data at cellular resolution. Okay, so lots of people are getting this data now. We started out quite a while ago, an embarrassingly long time ago, getting these data out of fly embryos. And so I won't talk about microscopy in any detail. Suffice it to say that we get this data with confocal fluorescent microscopy. You can look specifically for any kind of protein or RNA if you take the trouble to build the reagents you need. And so, for example, if you were staining for RNA from one of these reporter constructs, you get some picture that looks like this. Okay, these bright dots are nascent transcripts that are being transcribed right off the DNA, and if I have time, I'll show you a beautiful picture of those in live embryos from Mike Levine's lab. And then you computer process the data, and then what you can get is a quantitative map of the gene expression. And this is what you actually use your theoretical models on. This is the data set. Okay, and so what I'm showing you here is the distribution of these maternal genes I mentioned. Actually, two that I mentioned, bicoid and caudal. There are also other maternal genes that are present at relatively constant levels across the embryo. This is one called DSTAT that didn't emerge in the genetic analysis that I talked about earlier. This is the pattern of the gap genes, okay, hunchback, which is also maternal, although not shown up here, Krupel, Knurps, giant, hunchback again, and tailless. And this is this periodic pattern of Eve expression. Okay, now I want to introduce you to just one of the reasons why this problem is so hard. Okay, and this is something that in part came out of theoretical analysis, but I can explain it to you just by looking at the patterns. Okay, it's known from yes. Oh, I am so sorry. You know, I always tell my students, you know, if you put up a graph, say what the axes are, and, you know, I don't always obey it myself. The x-axis is the anterior-posterior distance along the embryo. Okay, so these segmentation genes, in most of the embryo, not counting the head, there's a beautiful Cartesian separation between genes that control the anterior-posterior axis and genes that control the dorsal-ventral axis. And the vertical axis is concentration. Okay? Now, it's known from multiple lines of evidence that the gap genes tend to repress even skipped. Okay, that's something that nobody disputes. And yet, if you stare at these patterns and say, okay, the genes in these patterns up here, which are rather broad, are making these patterns down here by repression, there's a certain point where you stare at the graphs and scratch your head and ask, how can this be happening? Okay, because there's something kind of crazy about it. Because look at Kruppel, it's really fat. Okay, underneath where the Kruppel domain is, there's a number of stripes. Okay, stripe 2 is sort of near the edge. Oh, by the way, I said there were 7 Eve stripes. All these graphs I show will only show 6 Eve stripes. And the reason is that stripe 1 is on the border of the head, and stripe 1 is a little bit puzzling. So I'll be focusing on stripes 2 to 7. Okay, so here's stripe 2 on the anterior edge of Kruppel. Here's stripe 3. Now what's happening here? It's coming up right in the middle of Kruppel. So if Kruppel is repressing Eve, how can that be? And more generally, you would expect that if Kruppel is a repressor, you should see a big hole in Eve expression here, and you don't see that. Okay, so there's something very tricky about this repression mechanism. Yes? This one, actually, these are all at the same time, okay, essentially at the time when this pattern of stripes have formed. And so that time is approximately 15 minutes before gastrulation. So pretty much everything I'm going to say today will be at this one time. And in the afternoon, I'll be talking about the dynamics of how the patterns change over time.
If they're all happening at the same time, then what's the cause and what's the effect?
Well, if you look over time, it is roughly true that the first pattern gives rise to the second, which gives rise to the third. And sometimes you'll hear people say that. Okay, it's actually a bit more intricate than that, because these patterns are constant or close to constant in time, so they essentially stay the same. They're providing an asymmetric background. Okay, and as I'll say in more detail in the afternoon, these patterns change in the sense that these genes come up in a localized way. This gene does not come up in a localized way. However, these domains, they get more concentrated. Okay, on the graph, they would grow taller. And as they grow taller, their repression kicks in and forces down these regions of non-expressing areas between the stripes. So there's a lot going on here. Okay, and what I'm focusing in on now is essentially the static picture of how the gap genes make these stripes to the DNA. Yes? Oh, yes, absolutely. Oh, yes. It's a snapshot in time, and there's extra information. Okay? Some of it is a little bit puzzling. For example, yeah.
Pattern A comes first, and then pattern B, and then pattern C?
It would be better to say that pattern A comes first and B and C develop together. Yes, A is laid out by the mother, and B and C are laid out by the zygote. So the problem is that if you can figure out some phenomenon using things from other chemistry and physics that you already know, that's obviously the way you should go. Okay, but if you don't, then you're in the tough position of having to figure things out. Okay, so that's essentially the design principles. Excuse me. The design principles for this model is that protein-DNA interactions are relatively well understood. Okay, so you can calculate them from thermodynamics. It's just a chemical binding problem. We use the approximation that the binding is in equilibrium. That's undoubtedly not strictly true, but it allows us to make good use of the PWMs. And it's approximately true. It's essentially saying that you're assuming that the binding of the proteins to the DNA is fast compared to the rate the patterns change. And then the sporty part is the protein-protein interactions, okay? Because there's two big problems. Number one, the chemical reactions are vectorial. And number two, it is certain that we do not know all the proteins that are involved, okay? This is a danger in doing this. I'm going to show you some successes of this model, okay? There's other problems we've worked on where it's obvious we need to know more. And so we're going to guess the form of the equations, but we're going to take various pieces of information in mind when making our guesses. The idea is not to guess carelessly, okay, but to think really deeply about different experiments, okay? And there's two key assumptions, two key things that we want the model to obey. Okay, one is, and this is closely related to this idea of canalization, it turns out that in these systems, the biological function, okay, that's a slightly fuzzy term. For those of you who are coming from physics, really, I'm afraid function means the thing about the system you happen to be interested in as a biologist, okay? That's vague, but that's what it is. In this case, we're interested in activation of repression in genes. Okay, the function resides in multiple sites, okay? And this is a very fundamental point about the difference between these systems and the bacterial system that Jacob and Monod worked on. In that system, there was a one-to-one mapping between chemistry and function because there was one single binding site that a protein bound to, and that was all it needed. Okay, one of the things that's very tricky about these systems is, you know, for example, that Kruppel affects Eve, but as you'll see later, there are lots and lots and lots of Kruppel binding sites, and changing one binding site will often have very little effect. So you have to write the equations so that you're taking into account point A, and then the other thing is that the activators, okay? And these genes seem to have the property that their default state is not to be expressed. To get strong repression, you need to have activators, and sometimes the action of these activators can be blocked. The activators can have really huge amounts of synergy. And when I say huge amounts of synergy, I mean, for example, you can do experiments with these genes in tissue culture, and you might find gene A increases transcription by a factor of 3, gene B increases transcription by a factor of 5. If you put A and B into the tissue culture cells together, you don't get activation by a factor of 8. That would be additive. You don't get activation by a factor of 15. That would be multiplicative. You might get activation by, say, a factor of 50. And the other reason these systems are tremendously complicated is that lots of different regulatory mechanisms exist, and typically they're all acting at once, okay? Now, This is a woefully incomplete list. Okay? It is, however, the absolute minimum that you need to describe the stripes of Eve. Okay, so some of these are very basic. Okay, protein binding to DNA, competition for bound sites. This falls out of very straightforward chemistry. Sometimes you have cooperative binding between proteins. And what that's saying is free energy of having two proteins bound is greater than the sum of the free energy of having either one bound separately. Okay, and what I'm about to tell you, the only cooperativity that we use is cooperative binding by bicoid. There undoubtedly are other cooperative interactions. We take a very minimalist view, okay, and that's important because if you're a theoretician and you want to make a new theory, okay, your first tendency is to think, oh, I want to make my theory completely general. It's got to be really general. I'm going to take everything into account. Okay, well, if you do that, that's a colossal error because what will happen is your theory will fit any possible data that you could possibly give it, and you won't learn anything from it. So you always sort of want to have your theory at the edge of what you can understand. And the idea is that then if something is wrong when you try to compare your theory to the data, it will be something that is wrong in a very limited way, and that will suggest one particular thing that you're missing. Now, there's two really critical things that you need. These are very fly-specific. Okay, you need short-range repression by quenching. Okay, and when I say short-range, I mean within, the repressor can affect the activator within about 150 base pairs. Now, the immediate thing that I know many of you want to ask is, what does that mean chemically? Okay, so I'm ashamed to say that I don't quite know, okay? One thing that's very suggestive is that 150 base pairs is about the size of the nucleosome. But when I said that you want to write these equations down based on phenomenology, what the phenomenology is you can do experiments where you have activator binding sites and repressor binding sites, and you vary the distance. And so from those experiments, it's quite clear that there's a fall off in repression for these repressors that have this property at about 100 base pairs, and their action falls to 0 at 150. Okay, remember I mentioned gap genes, those big fat genes, the genes that make those domains like Gaussians. Okay, they are all short-range repressors. Okay, and the short range is very important because this is what enables the enhancers to act separately. Okay, I said, you know, why doesn't Krupel just kill expression over a large range of the embryo? Well, the reason is that it can cause repression at one enhancer because there are Krupel binding sites there, but the other enhancers are more than 150 base pairs away, so it doesn't affect them. Okay, so this is now giving you the beginning of a functional reason for the existence of enhancers, but to check that, you have to make calculations. You still need a model. This idea was first proposed by Mike Levine on the basis of experimental data, but there's a lot of things going on at once. Okay, coactivation. Okay, I'll come back to coactivation in a little bit, but you also need this to understand the regulation of Eve, because what is coactivation? Okay, I told you that all the gap genes were short-range repressors. Hunchback is a gap gene. Okay, it's known from classical experiments in the Levine lab that Hunchback is an activator of stripe 2 and it's a repressor of stripe 3. This is crazy. How does it know what to do? It's not looking at a little GPS and seeing where it's binding. The answer is that other proteins can bind nearby and switch its regulatory sign. Okay, so for example, it's been shown very cleanly by tissue culture experiments that bicoid is a coactivator of Hunchback. So if there's a bicoid site within 150 base pairs of a hunchback site, it changes the regulatory sign of hunchback from negative to positive. Okay? And so this is why I said this was a minimum set of mechanisms for stripe 2. Direct repression is simply quenching near the basal promoter. If you have a quencher near the basal promoter, it can shut down the whole gene. This is really something you need that's an artifact of enhancer experiments, because the enhancers are not close to the transcription start site in the native situation. And in the absence of detailed chemical information, we model activation by a diffusion-limited Arrhenius rate law. So I'm sure you've all seen this before. This is the most mechanism-free model of a catalyzed reaction. Okay, the idea is that the reaction proceeds to the right. And it's thermodynamically favored by some delta G, but the rate of the reaction is controlled by an energy barrier here. And typically, well, the rate will then be proportional to e to the minus delta A over KT. And that would give you an exponentially increasing rate. Okay, but of course, the rate can't increase without limit, because ultimately new RNA polymerases have to come into the gene. Okay, so that'll cause it to flatten out, and so a diffusion-limited Arrhenius rate law will give you a sigmoid that looks something like this, 0 to 1 in arbitrary coordinates, where you get the synergy in this rising phase, okay, but then you hit a maximum and you get flatter. Okay, I see maybe I'm, well, let me not worry about how much time, and I can accelerate things in the afternoon. Okay, now look, this slide is kind of scary and ugly. I always like to show the model equations. Now, there's sort of a bad thing that happens, okay? There are lots of different mechanisms, which I outlined on the last slide. If you describe them mathematically, then you basically have lots and lots of different equations. And I apologize to my theoretician colleagues in the audience for the ugliness of this equation, okay, many, many, many years ago. But the problem is this is what the system requires, okay? And to use a great man's quotation in vain, the first time I ever gave an invited talk about this stuff, which was ages ago, Francis Crick was in the audience. And, you know, he said, first Mike Levine talked about transcription. There was no model then. And then I talked about the stuff I'm going to talk about in the afternoon. And Crick said, well, it used to be that if you had a theory that had more than 3 parameters, nobody would ever believe that it's right. But the system you showed, you know, it had 50 parameters. That's as many. You need 50 parameters to solve it. And this other stuff, he was talking about what I'm talking about today, you'd need 1,600 parameters. Okay, so fortunately that's not true because of PWMs, but I think this ugliness of a lot of parameters is something that you can't get away with, and one of the challenges for theoreticians is to find ways to take these big ugly models and simplify them and find ways to understand them better. Okay, you can get things out of the model that are much simpler than their raw form, but you have to go through this stage of complication because what's going on is very complicated, okay? And even though I'm sort of behindhand here, I am going to put in an editorial comment, okay, because I get to stand up here and talk to you, okay, where I respectfully disagree with some of my colleagues. Okay, many theoreticians believe that the most beautiful and elegant models have no parameters. Okay, for the purposes of biology, I think that that is incorrect. Okay, because although these theories are very beautiful, often, okay, what we want to understand is how organismal form and organismal evolution are controlled by DNA sequence. Okay, DNA sequence is a tremendous pile of parameters. And so if you want to get away from parameters, you're going to lose a lot. Okay, now what's in these equations? Okay, so parts of them I don't want to talk about. If you're interested, I did finally send reading material to VJ. I haven't checked if it's up yet. Okay, great. So some of these, so what we're looking at here is this is the paper that I sent for further reading because it has a probably excessively complete account of this model. So look, although this is a tremendously ugly slide in terms of mathematics, what's going on is very simple. Okay, it's a giant function. Okay, what goes in at the top are DNA sequences, positional weight matrices. Okay, those are essentially parameters, and concentrations of transacting factors. Okay, this will be covered, equation 2 will be covered later in the week. This is essentially how you get equilibrium binding constants K from positional weight matrix W. Okay, so you then have binding, if you identified the binding sites, each one of them has a equilibrium binding affinity, capital K, and what you're interested in is the fractional occupancy of each binding site. That's these Fs. Okay, you can write a general formula for this. The general formula looks extremely mathematically dense, but really when you're experienced with these things, if someone shows you a picture of a binding site, you can write down the formula for the fractional occupancy. The idea is if the site is 100% occupied, this F is equal to 1. If it's unoccupied, it's equal to 0. Okay, this V, superscript A, is the concentration of the transacting factor. Okay, this is a single binding site. This is two competing binding sites. And this is the case of a binding site that has coactivation going on. And then you take these Fs. What you're ultimately going to do is you're going to add up all the activators. Okay, the activators can act at a long distance from the promoter, but in order to do that, you have to do a few calculations first. And here's where the phenomenological guesswork happens. This is how you calculate coactivation, and I want to spend a few minutes on equation 7, which is the equation for short-range repression, because it gives you the spirit of how you can write down these phenomenological models. Okay, so you do essentially a gigantic convolution over the whole gene, such that for every binding site for an activator, that's this FA, you correct it for repression. And the way you do that correction is you take a gigantic product, okay, also over the whole gene, and you see that there are these, this big product looks like 1 minus Q of D times E sub BQ F sub QK. Okay, I'm not going to get into the notation, but it provides complete bookkeeping. It says where on the sequence the binding site is, what's binding to it, and then it gives it a name. This is repression by factor B. The A's and B's are which gene it is. Okay, so look at this factor. Okay, well, first of all, it has a fractional occupancy for the quencher. So if there's no quencher at the binding site, That'll make this product 0. You're just multiplying by 1. No effect. Just what you would expect. Okay, different quenchers have different strengths. So here's a parameter that says the strength of quencher B. And then what's this Q of D? Okay, this is the distance function. And so the idea is that this is equal to 1 up to 100 base pairs, and then it goes to 0 after 150 base pairs. So if you're more than 150 base pairs away, this quantity is 0. Again, you're multiplying by 1. So in an actual realistic case, if you look at how a gene gets repressed, okay, this is the phenomenological expression of the fact that you need multiple binding sites to have a single function. And so, for example, in particular cases that we've looked at, you often find that each activator is within range of 6 to 8 quenching sites. Each one provides a quenching of on the order of 0.6 or 0.7. That's really weak repression. But 0.6 to the 8th power is strong repression. Okay, so there's some chemistry we're losing, but we're expressing mathematically what we can see about how the system works biologically. So there's some free parameters here that yes. So I back up one. Yeah. You mean the sequence in between? Yes, I am implicitly assuming that. Well, not in the cases that I'm aware of that were how we set this up. Okay, it's quite possible that there are situations where that does happen, and it absolutely could happen in certain circumstances. For example, there are things called insulators that essentially control the range over which the enhancers act. So if you put an insulator in between, that'll change things radically. Okay, so the positional weight matrices are determined from independent experiments. And so, as I said earlier, the model is parameterized by genomic sequence. That's what we want. Okay, some of these phenomenological parameters, like strength of quenchers, we have to get by giant least squares fits, which you can do by either evolutionary computation or simulated annealing. Okay, these are large fits to data, but they're very well conditioned. This isn't like lasso. You have a lot more data than you have parameters. Wow, it's 10 to 11. Okay, let me tell you a little bit about an informative experiment. Okay, and this is the kind of thing, I have 2 informative experiments. I'll at least get through this one before the break. This is an experiment that's haunted me for 20 years, or more than that, a quarter century. Okay, so way back in 1993, this shows you non-quantitative data. Steve Small did an experiment where he took the 3+7 enhancer and the stripe 2 enhancer, and he showed that you could put them within about 300 base pairs of each other, and you get an essentially normal pattern. Okay, this is the expression of black C. Here's stripe 2, here's stripe 3, and here's stripe 7. But if you fuse them, you get a completely different pattern. Okay, you can see that fusing the enhancers in this non-quantitative data makes the stripe seem fused. If you look carefully, stripe 3 now looks a little fainter than stripe 2. So Aram Kim took this data and regenerated. The flies had been lost, but he reestablished the line. And he quantitated it. And actually, quantitatively, it's much more radical change than you see on the right. Okay, again, percent embryo length on the horizontal axis, RNA concentration on the vertical axis. When you fuse, the expression of stripe 2 goes up by a factor of 5, stripe 3 goes down, and the interstripe gets de-repressed, stripe 7 gets bigger. Okay, and by the way, you can make the fusion in the opposite orientation where stripe 2 is here, stripe 3 is there. In that case, stripe 2 gets really strongly repressed. Okay, so I'll tell you that this equation, that this experiment haunted me. Because my friends who are experimental biologists, they said, you know, John, why are you wasting time with all this mathematics when we already understand transcription quite well? And I'd say, well, fine. If you understand transcription, what is going on here? And so we modeled this, okay? And I don't want to take really much time on this slide. I'll simply say that this is the result of fitting the model to the fusion data. Okay, here you can see the repression of stripe 2 when you fuse in the opposite orientation. And the data are the dashed lines, and the model fit is the red line. Okay, and when you can't see the dashed line, it means they're right on top of each other. And so even though I gave 3 lectures about statistics last week, I just want to point out that we're looking for a lot more than statistical correlation here, okay? If you have a model like this, you really want to be able to fit the model to the data within the limits of your experimental error, okay? Because an organism has to develop, okay? Statistical correlation is not good enough. If it doesn't get things right, sufficiently right, it will die, okay? So in contrast to last week, I'm trying to get away from statistics here. Yes? Yes.
So how did you choose that time? And
well, this is basically the time when the stripes are fully mature. And so, look, I don't want to, this, you know, although I think this model is probably the best and the most predictive out there, I mean, I'm not unprejudiced, I want to share with you the fact that, and I'm talking mostly about things it gets right, okay? I want to share with you the fact that there are some things that it is not getting right. So we've only incorporated the structure of chromatin to a limited extent. And in our first investigations with this model, in a simpler case, it turned out that it worked perfectly well over time. But one of the experimental limitations of this system is that every time you take these artificial genes that you measure the enhancer activity with, and they tend to go in different places. Okay, so one of the problems with early experiments was they would go in random places in the genome, and that would have variation between them. So we have been very careful to use docking sites, except it turns out that the docking site we were using does not allow early expression. Okay, so there's plenty of mysterious things left to figure out.
Sorry, just a follow. So this is spatial pattern. That's
this is spatial pattern.
So do you have spatial diffusion in the model, or the spatial pattern is because of the spatial pattern?
In this model, there's no diffusion. So I show these graphs. Okay, I show these graphs because that's the way that we're used to thinking of it as fly biologists. You look at the embryos and you see stripes. But really, what you should think of these as is they are a series of about 65 assays of input and output. Okay? In other words, what I'll talk about this afternoon, which may spill into tomorrow, is a case where we're looking at how the patterns develop in time and we're taking diffusion into account. Okay, but here we're basically using the fly embryo like a microarray where we have the chromatin in the native situation, and each one of these data points, okay, goes from about 35 to 93% egg length. Okay, I haven't talked about how we get data, but it happens that each nucleus is almost exactly 1% egg length. So although this is average data, it's averaged in 1% bins. And so to a really a remarkably good level of approximation, what we're essentially saying is, this is what happens in nucleus 1, this is what happens in nucleus 2, this is what happens in nucleus 3, okay? To give you another sense of the intricacy of this problem, okay, remember that the gap gene patterns are themselves pretty intricate. Each one of these nuclei is seeing a slightly different combination of gap genes, okay? And I'm showing all of those assays simultaneously. You can sort of imagine the gap gene pattern superimposed on this. And if you're sufficiently masochistic as to want to read about this in detail, you'll find all the information there. Yes?
You could perhaps get the model to agree with the data by a whole, sort of multiple combinations of parameters. Is that correct, or is that is there a constraining of the parameters in such a way that you can reasonably?
Well, that's true, but not to the extent that it's a circular and useless enterprise. Okay, so for example, we're fitting on the order of 60 parameters to about 1,000 data points, and what essentially it turns out that you get is you get a cloud of regions where you have allowable parameters, but this cloud is actually relatively small. And as a sort of a practical matter of using the model, okay, the most frequent problem is that we give the model data that it's unable to fit, okay? And then if you have something that works, essentially what we do is we analyze specific cases of the model with slightly different parameters, and we make sure that the biological conclusions we have are shared in common, okay? Now, to do this even better, and some people have made some nice steps along the way, such as Doug Laufenberger and Johannes Jaeger, is that what you would really like to do is fully sample the distribution of allowable parameters. But we haven't really gotten into this because we've been able to draw biological conclusions without doing that. So that was maybe a more lengthy answer. You have a follow-up, Sanjay?
No, maybe we can take it up later.
Okay. I mean, essentially it works, and it's a little blurry around the edges, like a lot of things that one does with experimental data. So I have maybe 5 minutes. Okay, this is a school about evolution. So I do want to talk about evolution a little bit, but I think I'll have to finish what I'm talking about in the afternoon. Here's another informative experimental problem. Okay, so of course, these enhancers evolve. And this shows you the lineage of the Sophophora. That's the subgroup of Drosophilids that melanogaster's in. And you can see that here's melanogaster, and this shows their lineages. Okay, here's some other species, Jacuba and Erecta, Pseudo-obscura. Okay, these are all other species of Drosophila. Okay, so Misha Ludwig, who I actually now share a lab with along with Marty Kreitman, back starting in 1998, Misha isolated the striped 2 enhancers from these various different flies. Okay, and I would like to trace this back also to this idea I did call this lecture canalization. Okay, I said buffering of phenotype from genotype. Okay, well, this happens not only within species, but between species. So if you take these enhancers from these different species of flies and you express them in Drosophila melanogaster, they express, okay? And there is, in fact, an even more extreme example. And I think that this would be a good place to start in the afternoon because we had some questions. We want to start the next session on time. So look, I think I'm going to stop here. We have time for maybe 1 or 2 questions. And I'll continue this story in the afternoon. Thank you.
1 or 2 questions? Quick ones?
The transcription factors change their sign in presence of other transcription factors.
Yes?
How that happens? Like,
well, it's only partly understood, but the basic thing you should be thinking of, it gives me a reason to retrieve this piece of chalk before it's reduced to powder by someone's foot. The basic idea is that things get recruited. So if this is the DNA, and you have the transcription factors are binding to the DNA, you have other things which have various names, such as adapter factors, that bind to the transcription factor. Okay, now, essentially the idea of this coactivation is that what's probably happening is, if, here, I drew it without a coactivator. Adapter 1 binds and it makes repression. But essentially, if you had the coactivator bound nearby, and I'll write a coac here. Okay, then a different adapter factor binds and you get activation, okay? It's this business of building up the proteins in layers, okay? That's one of the reasons why the reactions are vectoral. And one of the frustrating things about all of this is we don't know enough about the chemical mechanisms.
So what I'm asking is like something like a chicken and egg problem. These transcription factors are basically DNA binding proteins, which have to be in turn transcribed from a DNA region, which in turn needs some other transcription factors. So which transcription factor comes first and make these transcription factors, which in turn make all these developmental
You're
changes.
talking about the fact that all these genes are interacting in some crazy way. I'm going to talk about that, although at the present moment, we coarse grain the model before considering that situation. Daniel,
Professor
you're sitting
Reinitz,
way up in the sky today.
so I understand that some error correcting codes have been proposed, right? So is that somehow incorporated in the regulatory mechanisms that you've proposed?
Well, yes, and that's the sense that I said canalization in cis for the title of the talk. Okay, because most changes, most base pair changes in the system don't affect expression much. And one of the reasons that the system was very hard to analyze is that when people isolated these enhancers, the first thing that they did was they tried mutating individual binding sites, and typically that had little effect or a small effect. Okay, the error correction in this case is sort of weak. Okay, I compared it to the law of large numbers. Okay, there is always it's really more buffering than active error correction. Okay, the next case I'll talk about with the whole network is error correction that involves the actual dissipation of energy. This is as buffering from the architecture of the system.